Hello everyone. Welcome to MRU E-Learning powered by Malla Reddy University. This is Dr. Divyabhandhavi Ravada, Assistant Professor, Department of Genetics and Plant Breeding, School of Agricultural Sciences, Malla Reddy University. Today, we are going to see the most fascinating topic of our molecular biology, that is CRISPR-Cas9. The CRISPR-Cas9, we will see the basic mechanism of CRISPR-Cas9 and also some of the applications in agriculture. The global agricultural production faces an acute challenge. By 2050. The world must feed 9.6 billion people, requiring a 60% rise in the staple crop yield. This is affected by the two major factors. One is the conventional breeding. There is a decrease in the conventional breeding strategies, and the next one is climate change. So the conventional strategies for developing new crop varieties are simply too laborious and time consuming. To overcome this, we have to depend upon the rapid sequencing technologies and genome editing technologies like CRISPR-Cas. So we know what is genome editing. The genome editing, it is a technology that precisely modifies the genome sequence to induce insertions, deletions, and base substitutions. We know what is an insertion. Insertion is nothing but addition of some of the nucleotides into the DNA strand. And deletions are nothing but removal of the nitrogen bases from the DNA, and base substitutions are nothing but we have to substitute the adenine, guanine, cytosine, thymine. So we know that the base DNA structure will have four types of nitrogen bases. One is adenine, thymine, guanine, and cytosine. So among these, adenine and guanine are the purines, thymine and cytosines are the pyrimidines. In a DNA structure, adenine pairs with the thymine with a double bond, and guanine pairs with the cytosine with a triple bond. Before the genome editing technologies, before the CRISPR-Cas, we have few more genome-specific genome editing technologies. Those are the meganucleases, zinc finger nucleases, and tellins. So tellins are nothing but transcriptional activator like effector nucleases. But these three genome editing technologies construction requires a complex protein engineering. So because of this complexity, their usage is become limited. So we should depend upon the CRISPR-Cas9 technology. CRISPR is nothing but clustered, regularly interspaced, short, palindromic repeats. These are associated with the Cas9s. This CRISPR-Cas9, it is originating from a prokaryotic immune system, bacteria, and this by using this CRISPR-Cas9, we can modify the plant genomes with exceptional precision, efficiency, and programmability. This technology have been utilized in the diverse crop species to enhance yield, for disease resistance, and for quality, and also for the stress tolerance. This CRISPR-Cas9 also helped in the domestication of the wild species, in advancement in biotechnology, including gene regulation and protein engineering. Finally, the CRISPR-Cas9 stands as a transforming tool for sustainable agricultural and global food security. So coming to the CRISPR-Cas9 basic thing, this CRISPR-Cas9 can destroy the targeted DNA at specific locations. Here we can see this is a targeted DNA. This CRISPR-Cas9 technology can break this targeted DNA at the specific sites. So CRISPR, what is CRISPR? CRISPR is nothing but a short RNA, and Cas9, it is a protein having nuclease activity. So these two components of CRISPR are CRISPR and Cas9. So CRISPR should match with the targeted DNA, and the Cas9 will come and cut at the specific locations. The basic concept behind this CRISPR-Cas9, this Cas9 depends upon the idea of bacterial acquired immunity. So we know that bacteria sometimes infected by this virus. The virus which attacks bacteria is called as bacteriophage. So basically the bacteria utilizes this CRISPR-Cas9 technology to protect itself from this phage virus. So when phage attacks the bacterial cell, the phage will transform its genome into the bacteria. So we know that the bacteria have a tendency of any foreign material comes, the bacterial genome will go and attach to it. Finally, the bacterial genome get attached with this viral genome. The phage will have CRISPR kind of sequences. This phage genome will have CRISPR kind of sequences The phase may destroy the bacteria, or the bacteria may survive after the viral attack. If the bacteria survives, it will store the genome as a memory and then by using that memory, the bacteria prepares a RNA copy along with that Cas9 protein. So whenever in the future, if the same type of virus attacks the bacteria, and the bacteria phase releases its genome content into the bacteria will identify that is by using its past memory, it releases this RNA copy along with the Cas9. This RNA copy from the bacteria, it will go and attaches to the viral genome. This is here we can see these scissors, molecular scissors are nothing but this Cas9 protein. The Cas9 protein along with that RNA sequence. This is the RNA sequence. This complex will go and attaches to this viral genome. After the RNA get paired with the genome of virus, then the Cas9 will come and cuts the viral genome. So this is the basic concept behind the CRISPR-Cas9. This is a technology. The bacteria utilizes this technology to get protected from this virus. So same technology now we were doing in the vitro conditions. So let's see the mechanism. So in this CRISPR-Cas9 mechanism, there are two methods will be there. One method, the CRISPR RNA with the Cas9 or CRISPR RNA with the Cas9 along with this trans-TR-CR RNA. So now we will see this is a targeted DNA, this blue strand and this green strand and blue strand are the targeted DNA. The CRISPR along with this Cas9 protein, it helps for the cleavage on the double-stranded DNA. So this pink colored one is the CRISPR RNA, and this blue colored strand, the DNA strand, and this again green color and blue color strands of DNA will have some sites for this DNA cleavage. Those sites are called as PAM sites. And we should also know that what are these NGZ? NGZ are the special sites which are presented at the PAM sites. So the PAM sites are nothing but protospacer adjacent motifs. So what are protospacer adjacent motifs? For that we should know what is a spacer. The spacer is nothing but this is our CRISPR RNA. The CRISPR RNA will have complementary sequence with this targeted DNA. So whatever the portion of this CRISPR RNA having complementary base pair with the targeted DNA, this portion is called as spacer. The strand of DNA, the targeted strand of DNA which is having complementary with this spacer is called as protospacer. The place which is adjacent to this protospacer and spacer is called as PAM sites. These are protospacer adjacent motifs. And one more thing here we should know, what is this TR-CR RNA? TR-CR RNA is nothing but this is the extended loop of this CRISPR RNA. The main function of TR-CR RNA is to stabilize the CRISPR RNA. This TR-CR after attachment of this CRISPR RNA with this DNA targeted sequence of DNA, then this TR-CR RNA get deleted from this CRISPR RNA by the help of RNase 3. This RNase 3 will cleave the loop between this CRISPR RNA and TR-CR RNA. So this is the main mechanism. Once this CRISPR RNA get attached with the DNA targeted DNA, then the PAM sites will get activated. Then after attachment of this, when the CRISPR RNA attached with this main DNA strand, the Cas9 will come and attaches to the targeted site. After that, once the Cas9 is get attached with the targeted site, the PAM will get activated. Once the PAM is activated, then the NGG sites will trigger. These NGG sites after triggering, this Cas9 will go and cleave at the sites of NGG. So then we can see the double strand break. So these double strand breaks are of two types. One is non-homologous end joining. Some cases, these are the homology directed repeat. So non-homologous end joining repeats, there is no requirement of, this is a basic innate repair mechanism produced within the organism body. So whenever this NH is a non-homologous end joining repeats, this end joining occurs, there is no involvement of any template DNA, but homology directed repeat, we need one template DNA for its further attachment of the double strand. So gene targeting in plants. The gene targeting plants mainly depends upon the process called homology directed repeat. This is HDR, but homology directed repeat is not very efficient in plants, which makes it difficult to use widely. To overcome these problems, the scientists have developed one new genome editing technology that should not rely on this HDR. For that, they have given the base editing and prime editing. After the success of cytosine base editing and adenine base editing, in the human cells, scientists adopted the systems for plants. Those are the dual base editing. Another one is base editing based DNA deletion methods. Next, first we will see what is cytosine base editor. So the name itself says that cytosine base editor is nothing but it is a challenge. The DNA base pair will converting from the CG to TA. We know that. And A pairs with a T with a double bond. C pairs with a G with a triple bond. So in this cytosine base editor, it is used to modify it has used the modified version of the Cas9. So basic Cas9 is different from this Cas9. This is called as N-Cas9, having a mutation of D10A mutation, which can break at the one DNA strand. So basically the Cas9 will go attaches and it will cleaves the the two strands of the DNA get break from this Cas9. But in case of this modified Cas9 called as Cas9 with a D10A mutation, it will cleave at the single strand of DNA. So in these cytosine base editors, we will use cytosine deaminase is the one component. Another component is UZ1. UZ1 is uracil glycosylase inhibitor. What is the function of cytosine deaminase? This changes cytosine in DNA into the uracil. So this will convert C into the uracil, and this UG1 uracil UZI inhibitor, uracil glycosylase inhibitor, it protects the uracil from being removed from the cell's repair system. So by seeing this conversion of C to U, the cell repair mechanism will get triggered and it will see that there is error has been occurred. Then that UZI, it will help for the protection of this U. It will not allow U to change. The cell should not change this U. This N-Cas9 is guided to a specific DNA sequence by this sgRNA. So here the main component is cytosine deaminase and N-Cas9. The cytosine deaminase changes the cytosine to the uracil in the exposure strand. So this is a strand, the double-stranded DNA, where we have to do the modifications. In this, the exposure strand first attaches to the CRISPR-RNA. The first attaches to this CRISPR-RNA. Then this will change, the cytosine deaminase changes C to U. Then the UZI blocks the cell from removing U. Then N-Cas9 nicks the opposite strand and that triggers the cell for DNA repair process. By seeing the U and G mismatch, the cells will change the nitrogen bases into the T and A after the DNA repair. Finally, by using the cytosine base editor, the specific DNA base pairs, the cell from CG2, it is converting into the T and A. The next one is adenine base editing. So adenine base editing is similar as a cytosine base editing. In cytosine base editing, we are using cytosine deaminase. Here we will use adenosine deaminase. Okay. And one more is Cas9 protein. This is a modified version of the Cas9 protein with the D10A mutations. This enzyme changes adenine into the inosine. And during the DNA repairs, the cell reads this inosine the cell reads inosine as a guanine. In this A-T pair is converted into the G-C pair. But ABE, adenosine base editors, are not work efficiently in the plants. To improve this efficiency, the scientists are using some of the strategies. The one is the 3SV40 nuclear localization signals, and another one is modifying the sgRNA scaffolds. Another is simpler and single version of the enzymes. By these three modifications, they are efficiently using the adenine base editors in plants. Next, we will move into the dual base editing. So in the previous cytosine base editing, adenine base editing, we are converting the single base from C to G into A to T or A to T into the C to G. But in dual base editing, at the same time, two types of DNA changes can be happened in the plant, either C to G into T to A or A to T into the C to G. This is done by using one single sgRNA which guides the editing process. So in this we will use cytosine deaminase and adenosine deaminase. Along with that, we have to use this N-Cas9 with D10A mutations. In UZI, uracil glycosylase inhibitor prevents the cell from undoing the C to U changes. This combined system is called as STEAM. This is saturated, targeted, endogenous mutagenesis editors. This is called as STEAM, and this creates multiple mutations at one time. It can modify the regulatory DNA regions, and it enables large scale high throughput genome screening in the plants. Okay, next we'll move to the CBA based precise DNA deletions. So deletion means nothing but this removal of the nitrogen bases from the DNA strand is called as deletion. So now we are using the cytosine base editing precise DNA deletions. So in this simple it is similar like the cytosine base editors, the cytosine into the uracil. So normally the enzyme, we have seen that UGI, this is the enzyme which protects the uracil. It protects as a shield to the uracil to protect the cell to remove this uracil. But however, when UDG is overexpressed, so how come this UGI is working? This will mask the action of UDG. So UDG was the one which removes the mismatch of urine. But UGI protects the U by masking the UDG. Whenever there is overexpression of UDG, that removes urine and creates a special DNA sites called as AP sites. Those AP sites are apurinic, apyrimidic sites. There is no purine, there is no pyrimidine. These sites can then be cut by this AP lysase. If this nick happens near to the double strand break, so if this nick happens near to the double strand break, it may causes the deletion of this specific DNA sequences, specific nitrogen bases. Based on this idea, the scientists have developed new technologies called as APO-BEC Cas9 fusion-induced deletion systems of it. This fusion-induced deletion systems, the scientist has given this system. So in this, the most important things are this cytosine deaminase, Cas9s, UDG for removal of this U bases, and AP lyase. The AP lyase will cut the AP sites. The next important thing is prime editing. While cytosine base editors and adenine base editors can make precise base transitions, they cannot create all types of base changes. To overcome this situation, scientists have developed the new genome editing technology in 2019 called as prime editing. So in this, almost all 12 possible base substitutions we can do, and precise insertion up to the 44 base pairs and precise deletion up to 80 base pairs we can do, or even combination of these editings also we can do by using this prime editing technology. So now we will look into some of the applications in agriculture with the CRISPR-Cas9. The first CRISPR-Cas9 mediated molecular breeding accelerates the crop yield. So in case of rice, the gene which is responsible for this activation of the cytokinin, that is LOS-L5 gene. In case of rice, this gene, by using the CRISPR-Cas9, we are targeting this gene. This help in the activation of cytokinins. We know that the cytokinin is helpful for this tillering capacity in case of rice. So that the more tiller leads to the more grain yield. So we are targeting gene is LOGL5 gene. In case of wheat, CKX gene, which breaks down the cytokinin. The cytokinin in case of wheat, it helps for this senescence. So by breaking down the cytosine in case of wheat, we are expecting the more yield in case of wheat. Next example is rice by AAP3 gene. This is the amino acid permease 3, which is responsible for this nutrient distribution. And one more gene in case of rice is PIN5B gene, which controls the panicle size. In rice and wheat, GS3 gene, which controls the grain size. And in rice and wheat, two more genes, GW2 and ZW5, which controls the grain weight. By using this CRISPR-Cas9 mechanisms, we are accelerating the crop yield with these applications. CRISPR-Cas9 mediated molecular breeding accelerates crop quality also. Coming to the crop quality, if you see, when we compare the crop quality is determined these two factors. One is external factors, another one is internal factors. The external quality attributes are nothing but size, color, shape and fragrance. And this contrast, the internal quality parameters are this protein content, starch content, lipid content, and carotenoids, lycopenes, and flavonoids, and so on. So by using this CRISPR-Cas9 technology, we are not only improving the quality in the phenotypic quality as well as this internal quality also we are changing, like amylase content, enriched nutrient, carotenoid content, GABA content, and reduced gluten content in wheat, and reduced anti-nutrient contents. So we'll see the examples now. So by knocking means the expression, we are stopping the expression of gene GBSSI, that is amylose synthesis gene. We can generate the Paxi maize, which having low amylose content for better cooking. Not only in maize, we are using the same technology in rice also for decreasing the amylose content. And next is starch branching enzyme, that is amylopectin created high amylose rice. So very beneficial for this human health. And few more examples in case of wheat. And in wheat, the gluten loci created the low gluten content in wheat, and carotenoid content and GABA contents and nutrient enrichment also we can do. And in some cases, by using this CRISPR-Cas9, we are decreasing the phytic acid content, mineral absorption content in some of the phytic acid content in some of the crops. Fatty acid profile also, edited genes for high oleic acid content for healthier oils. So these are the things we made by using the CRISPR-Cas9. So one more application in agriculture, CRISPR in disease resistance. So instead of adding resistant genes, the CRISPR-Cas can disable plant susceptible genes. So here what we are doing, we are masking the susceptible genes, offering a safer example in the crops. First we'll go with this most important crop, that is rice. Varieties sativa. Bacterial blight. We know this bacterial blight is a most devastating problem in case of rice caused by Xanthomonas varaceae. So whenever the rice plant is get affected by this Xanthomonas species for bacterial blight, this will trigger the sweet genes. The sweet genes will release some products. Those products, because of those byproducts, the plants become susceptible for this Xanthomonas species. So in this technology, by editing the sweet 11, 13, 14 promoters, by editing these 3 promoters, we are going to get the rice plant having resistance to this Xanthomonas varaceae. Next example is citrus, editing LOB1 promoter resistance to the Xanthomonas citrae, that is citrus canker. In case of wheat, mutation of EDR1 MLO genes, resistance for this powdery mildew. So same like bacteria, whenever the bacteria faces the virus, that is bacteriophage, the bacteria uses the Cas9 technology to kill this virus. So for viral resistance, We are using this CRISPR-Cas9 technology to protect the plants from Gemini virus and Colimovirus. Knocking out susceptible genes can give broad virus resistance in case of against the 40 viruses. So these are the applications for this disease resistance. Now we'll move into the herbicidal tolerance. So nowadays in the agriculture, the weed problem also is the most increasing day by day. This weeds also decreasing the crop yields and also the soil health. So developing herbicide resistant crop is cost effective and eco-friendly solution for this. So in this case, we have to target the genes which are responsible for this herbicide resistance. In ALS gene is acetolactate synthase gene, which is targeted for herbicides like sulfonyl urea. The sulfonyl urea stops the activation of acetolactate synthase. So base editing in rice, in case of rice, we are getting the herbicide resistant plants by this ALS gene. And next example for this is ACCase, acetyl-CoA carboxylase. This is important for this lipid biosynthesis. So this gene is edited in case of rice and wheat. This is tolerant to the haloxyphop and quasilophop. Another examples for this is EPSPS. We know that shikimate pathway, the resistance for this glyphosate, and PPO, polyphenol oxidase, that is also having this gene also helps in the tolerance for this beta-phenyl herbicide. So applications in breeding technologies. So till now we have seen the applications in this different like disease resistance, herbicidal resistance, and quality and yield oriented things. Applications in breeding technologies also we have. Those are haploid induction, generating male sterile lines, fixation of hybrid vigors, and manipulating the self-compatibility. By these applications, we can increase the crop improvement. This technology is also, this CRISPR-Cas is also helping in improvement of these technologies for improved crop yield. The conclusion for this is CRISPR-Cas9, it is a precise, simple and cost-effective method revolutionizing the plant breeding and functional genomics. It is the most popular and adaptive gene editing systems. It enables the rapid development of new crops and it also helps in the increasing yield, nutritional value and resistant to the biotic and abiotic stresses. The applications ranges from creating low gluten wheat and non-browning bananas to improving the nutritional profile of staples like rice and tomatoes. So here about this basic mechanism of CRISPR-Cas9 and few of the applications in agriculture. So thank you so much for watching. Thank you.